In the earth sciences, we can study a lot of different things that shape the world around us, and many of us use what we learn to help people in our communities. And an earth scientist can investigate almost anything: volcanoes, the climate, tornadoes, water, earthquakes, rocks, the ocean, and even other planets like Mars. Today, we're going to focus on the branch of earth science called paleontology, and I'll show you some of the fossils they might study and what those fossils can tell us about what Earth used to look like. So to start with, fossils. What are they and who studies them? A paleontologist is someone who studies the history of life on Earth using the fossil record. And a fossil is any evidence left behind by a living thing. Fossils can be bones or shells, or carbonized plants, or even bacteria. Fossils are preserved in different ways too. Some fossils only leave behind an imprint in the mud, which means that even old dinosaur footprints are fossils. Bone can be broken pretty easily, and after something dies, its bones can crumble, leaving nothing for a paleontologist to find. But some bones were remineralized and become much harder as fossils, which is why we might find them buried in sediment and rock hundreds of millions of years later. But finding fossils is challenging. Where are they? You can't just put a shovel into the ground anywhere and expect to uncover something. And even if you do find a fossil, the recovery process can be intensive and you risk damaging it while you dig it out of the ground. And even then, if the fossil is removed safely, sometimes they're never displayed because leaving them out for viewing could damage them too. But let's say you're determined to go out and make a great discovery. Well, hold on a second, because paleontologists first do a lot of research about where they're going to go. The location depends on what kind of fossil you're looking for. For example, I wouldn't go out looking for a velociraptor fossil in Tennessee, but some incredible dinosaur fossils have been found in Utah. Once you have your location, then it's time to start digging.
We came out to excavate a sauropod this year, and we thought it would be a great specimen for the museum. So day one, we haul a lot of gear out to the site. We haul picks and shovels, and it's amazing. In paleontology, we use a range from a dental pick all the way up to a 60-pound jackhammer, and sometimes we flip between those pretty rapidly. So we'll be jackhammering for a day, we'll take off all the overburden, we'll be crack hammering with giant chisels, we'll find a bone, everybody slows down.
We use smaller hammers, we use finer chisels to remove rock that's close to the bone so that we can see what's actually in the quarry itself. And we don't want to use a big pickaxe, right next to the actual fossil because you don't want to damage anything.
Paleontologists often have to jacket the fossil during the digging and removal. They slowly dig as close as they can to the fossil, but then they will coat the rest with plaster so it can be safely transported back to a laboratory.
When we're getting a specimen ready for transport, we can't just take it out of the ground and put it in a truck and bring it back because it's too fragile. It needs some sort of cradling and some support. We uncover the surface of the bone and leave it in the ground. We don't take it out of the ground right away. We'll coat the surface of the bone that's exposed with a protective layer of toilet paper, or sometimes we use paper towels. And that will keep the plaster from sticking directly to the bone.
And then we'll mix up some plaster in water and cut burlap strips. We'll dunk those in and cover that whole specimen encased in rock and plaster, and we'll undercut it and flip it over and jacket the other side, and it's all ready to go back to the museum.
Once the fossil is removed from the rock, the next step is to catalog it. This happens once everything is brought back to the laboratory. Most information about a fossil is discovered in the lab. It's not the most glamorous part of the science, but it can be exciting in its own way. Once we know what the fossil is and all the information is properly recorded, the fossil is cataloged and stored or prepared for display. Many fossils are never displayed to the public because they're too fragile, in which case we might make a replica to display instead. So what kind of information does a paleontologist gather from studying fossils anyway? One thing they look at is the rock in which the fossil was found. The rock that surrounded and buried the animal or plant tells us about the natural environment where the organism died. For example, if the rock is made up of very tiny mud particles, the organism, like this fish fossil, would have died and been buried in a deep ocean or lake where the water is calm and fine sediment can settle around the fish. But if the rock is made up of bigger sand particles, gravels, or pebbles, then we can deduce that the organism, like this T-Rex, probably died on land or in a river environment where the wind or water has strong currents and covers the organism with large grains. Carl Linnaeus developed the modern-day system for how we identify and classify living things. We call it the Linnean taxonomic classification system, and we use this system to group together similar types of plants and animals. We can apply it to fossils as well. Taxonomy is the branch of earth science that deals with the classification of organisms, living and dead. And so when we talk about fossils, we will be looking at different taxonomies, or to put it another way, different species. All life on this planet, past and present, is split into three main domains: bacteria, archaea, and eukarya. To further classify eukarya, we use the 7 different groups that Carl Linnaeus developed. The largest subdivision is a kingdom, and one kingdom includes all animals on the planet. Another kingdom is all the plants, and another is all fungus, like mushrooms. After that, we split a kingdom into different phyla, then a phylum into different classes, then orders and families, and then genus, and finally, different species. There's a phrase you can remember, used to remember the order of all 7 subdivisions. If we look at the first letter of each one, K for kingdom, P, C, O, F, G, and S. And if we make up words for each letter, we get king for K, Philip came over for good soup. King Philip came over for good soup. So maybe that can help you remember the order of all of those different classifications. But remember, we use these groups to classify different organisms. So let's try a few. Let's start with people, humans. We're living things and we're animals. So we belong in the Animalia kingdom. That's for animals. Our phylum is Chordata because we have spinal cords. And then there's the mammal class, primates order, hominid family. Our genus is Homo, and our species is sapiens, or Homo sapiens. The species is always written with the initial of the genus first, H sapiens, or Homo sapiens. And that's us. That's our classification. Let's do another example. What about chimpanzees? We all know what chimps look like, right? Here's a picture of one. Now, chimpanzees share a lot of the same groups as us for their classification. In fact, the first 5 are all the same: animals, chordates, mammals, primates, and hominids. However, we belong to a different genus and species. Humans are Homo sapiens, but chimpanzees belong to the Pan genus and the troglodyte species. So while we are a little similar, humans still have some pretty important differences as a unique species. Okay, now what about dogs? Most people like dogs. They're our 4-legged furry friends after all. Dogs share 3 of the same classification groups as humans and chimpanzees, the kingdom, phylum, and class. But the last 4 parts of their classification are different, giving us the genus and species of Canis lupus for dogs, or C. lupus. All right, one last one. Let's look at clams. You can't get any different from humans than clams, except we still share one thing in common. Humans and clams are both animals. So we are still in the same kingdom together, the Animalia kingdom. However, we are different in all the other categories. For instance, clams are mollusks and bivalves, and they live inside shells. Humans have spines and brains, and we live on land. Now, lastly, I want us to look at some pictures of actual fossils, and I'll show you some of the things we know about them. Remember, we classify all these fossils based on the taxonomy groups I just talked about. And all of these fossils are organisms that lived in the ocean. They're pretty common and are fossils you could find outside if you know where to look. This is not just because life began in the oceans, but because shells and exoskeletons become fossils more easily than soft parts, like skin or leaves from plants. Now, let's take a look at some of these fossils of ancient ocean animals. First are the trilobites. Trilobites belong to the Arthropoda phylum, the same phylum to which spiders, centipedes, lobsters, and other insects and crustaceans belong, which is maybe why some people describe the trilobite as a giant cockroach that lived underwater on the sea floor. But don't worry about seeing these arthropods if you swim in the ocean today. They went extinct 250 million years ago. Here we're in a different phylum than the trilobites. This phylum is for mollusks. There are a lot of different types of mollusks, but these shelled organisms belong to the Pelecypoda class. The pelecypods, or bivalves, have been around since the Cambrian, like trilobites, but many of their species have survived to present day. These are animals like clams, oysters, mussels, and scallops. Have you ever seen a clam walk? This GIF is actually a scallop, but clams and mussels move in similar ways. What looks like a tongue is the animal itself, and that part of it is called a foot. A clam or scallop uses the foot to push itself away from danger and to dig into the sand and bury itself for protection. Those are pelecypods. Ammonites are mollusks, like the clams and oysters, but ammonites belong to the cephalopod class rather than pelecypod. Ammonites have spiral shells like some types of snails, but the animal looks more like a squid or an octopus, which are also part of the cephalopod class. Now, ammonites may be extinct, but one of their close relatives is still alive. It's called a nautilus. And although its shell looks similar to the ammonite, the nautilus actually belongs to a different class that we're not going to talk about today. Echinoderms include starfish and sand dollars and sea urchins, which eat microscopic organisms called plankton that float through the water. Their bristles catch the tiny plankton and move them towards the center of its body, which is its mouth, on its underside. Crinoids here is another species of echinoderms. Crinoids have been around since the Cambrian period, and some of their species are still around today. All crinoids have fine feather-like feeders or arms that capture plankton, much like the starfish and sea urchin. The main predator of crinoids are sea urchins, which can eat the tentacle-like arms of the crinoid. But luckily, the crinoid can regrow those parts back. And while the crinoid might look like a plant with a stem and a feathery crown, it is very much an animal in the Animalia kingdom. So those were just some of the fossils you will find today as you dig through the sand that is on the tables here. Everyone will search through 3 dig sites. One with very old fossils from the Paleozoic, one with fossils from the Mesozoic, and one dig site with fossils from the most recent era, the Cenozoic. As a junior paleontologist, you will dig very carefully using your hands to find the buried fossils. Remember, fossils are fragile, and paleontologists are always gentle when searching for and removing fossils from the ground. As you find the fossils, identify them by matching them to the pictures at your dig site. And once you find all the fossils in the sand, you will answer some questions about them. Work as a team and write down what you think are the best answers. Then put all the fossils back in the sand and carefully bury them again. When everyone is ready, you'll move to the next dig site where you can begin searching for new fossils. And remember to take your answer sheets with you.